Okay, so in our next chapter, we'll be looking at a specific instrument, and then this instrument is very key in, the life of a scientist. Now, by the end of this video, you get to know the type of instrument I'm talking about. Maybe in the video too, I'll mention it. So you stay tuned. All right. So in the early days between the 13th century and then the 16th century, we were having these fleas and then the spectacles. The fleas are spelled Fleas, and then spectacles. So you see that some of the scientists, they will just bring down something and then they'll use their specs to do this way in some acrobatic way, just to be viewing microorganisms. Yes. But in this process, it was only one lens. It was only one lens that makes it a simple lens. Remember, we have two types of microscope. We have the compound microscope and then the simple microscope. The simple microscope are the lenses. Then the compound microscopes are the microscopes with two glasses. So in the early days, we were just using the simple microscope. Now, in 1590s, there was a breakthrough. And then now this breakthrough happens because a father and son combined their head and then brought up a compound microscope. The name of these scientists were Hans and Zacharias Janssen. Now, they experimented with multiple lenses in a tube. They discovered that an object at the end of a tube appears significantly larger than with a simple magnifying glass. This was the birth of a compound microscope. So in this process, they had a tube, fixed one lens at the top, fixed one lens at the bottom, and then they were viewing through it. And then in their observation, they realized that as they are viewing through this lens, the object appears to be larger than the tube in which, or the magnifying glass that contains only one lens. Okay, now fast forward, we move to the golden age. So in the golden age, that is around the 17th century, perhaps nobody was born. Yeah. So in the 17th century, this is when the microscope moved from a toy to a more serious scientific. So at first, they were seeing the microscope to be a toy, to be like, how should I say? They first write it as a novelty. So they will just write a book, then talk about it. This is a microscope. People will be reading about it, but it wasn't that useful until in the golden age, they tried using the microscope to view tiny organisms, and then they were realizing how serious or how powerful the instrument is. So I hope we've all heard this name before. Antoine van Leeuwenhoek, that scientist, he's very common in our books. He is the father of microbiology, you know that. Yes. Now, Antoine van Leeuwenhoek wasn't a trained scientist, as you know. He was a fabric merchant who wanted to see the quality of his thread. So a fabric merchant, are the people, let's say the seamstress and then the tailors, and then those who that wave kente and stuff. You get it? Yeah. So he wanted to see the quality of his thread. So he created tiny high quality single lens that could magnify up to 270 times. You know, times in our previous video, we did the times. That means magnification, right? So he was the first to see animalculus. When we say animalculus, it means bacteria and then protozoans. Now, he was the first to see animalculus in a drop of pond water because he was able to use the compound microscope to view like 270 times. So he is a genius. So after father and son combined, bringing up the compound microscope, Antoine van Leeuwenhoek, who was a fabric merchant, also tried to, view tiny thread or the quality of the thread. So he used this microscope to view object 270 times. And then in that process, he was able to discover animalculus, bacteria, and then protozoa, which have been found in water or pond. Okay. Now, there came one guy. That one guy is very crucial. That one guy is called Robert Hooke. Others will say Robert Hooke, but please, it is Robert Hooke. Yes. So the Hooke is spelled Hooke. Okay. But so far as it is Hooke, doesn't mean you should pronounce it Robert Hooke. It is Robert Hooke. Now, Robert Hooke was naming the cells. So for him, he was just observing a thin sliced cork, and then using a microscope. And then he said that how compact these cells are. It's like a room. And then he named the cells. So in 1665, this Englishman, Robert Hooke, published a micrographia. Now, a micrographia is like, say, a graphic, and then it has been, published, or a book that has been published. Now, using a compound microscope, he looked at a thin slice of cork. He saw tiny walled compartment that reminded him of a small room monks live in. So as he was using the microscope to view a thin slice cork, he saw a tiny, boxes that reminded him of the rooms the monks have been living in. Those small rooms. And then in that rooms, they call them cells. So like you go to the prison, we have small rooms, and then those small rooms, they call them cells. And then Robert Hooke said that, let me call these rooms I am seeing in my microscope cells. And then the name stuck. So imagine I was Robert Hooke, and then I was looking through this microscope, and then I observed a thin sliced cork, and then it was smaller. How will I call it in my language? So try sending me a message in the comment section telling us about how you call a small room in your language. Thank you. Bye-bye.